Hi, my name is Eric Weingart, and this PowerPoint is over my essay that I just wrote regarding the protein tubulin.
This PowerPoint is over the protein tubulin. When we think about proteins, we think about food and how we need to eat protein to live. Proteins are essential for life, but that's not the kind of protein that we're talking about. Proteins come in all different conformations. To the right, we see the most abundant protein in the human body. The structure of this protein determines its function, and proteins function to keep us alive. The genome within our cells is what helps to code for these proteins. These genes are the instructions. And so we see these base pairs in the DNA, and these base pairs are going to create the amino acids that combine together to create these different proteins. And we see this green, this red, and this blue protein alone. And they can work alone, but they can also form these complexes to perform other cellular functions. Some of those functions can be to respond to stimuli, catalyze reactions, synthesize and repair DNA, transport molecules across plasma membranes. They can work as hormones or enzymes, and they provide structure and support. So we see in this image there's a transporter protein moving molecules across this membrane, receptor and enzyme proteins on this membrane to catalyze reactions or start reactions to tell the cell to grow and divide. There are anchor proteins for structural support, and some of these protein structures are going to be, well, they're all three-dimensional, and that is because they fold and covalently bond with other molecules in that structure. And then there's 4 levels to those protein structures, which include primary, secondary, tertiary, and quaternary. Not all proteins have quaternary structure, but most of them do. Every protein is made of an amino acid sequence, and this forms polypeptide chains, which we know to be primary structures. Secondary structures include a broader range of structural types, such as alpha helices and beta strands. This alpha helix structure forms a spiral, and in this image we can see as a secondary structure the spiral. And if we want to ignore that for a second, we can move on to beta strands. Beta strands are very linear, and when they hydrogen bond to other beta strands, they form what is known as a beta sheet. And so if we look at this image, it looks very linear. And so when we move on to the tertiary structure, we see only these beta sheets. And so these tertiary structures are also called domains, and they're really just these structural elements packed together. And when you pack all these together and you have multiple tertiary units and you put all the tertiary units together, we form quaternary structures. So tubulin is a structural protein. It will form these microtubules, as we see in the image to the right. And tubulin functions to regulate those microtubules that provide structure and stability to cells. There are 6 different conformations of tubulin. They exist, not all of them, but most of them, at least one form in all eukaryotes. And this is because as long as they have a cytoskeleton, it'll exist within that organism. And tubulin is not in prokaryotes that we know of. However, we do know that there are microtubule-like structures, so there has to be some related protein. There are 5 types of tubulin in humans, and alpha and beta tubulin are what make up those microtubules in the cytoskeletons. Beta tubulin exclusively forms the neurons in human brains. In this image, we see just how highly expressed tubulin is in the brain compared to other organs in the body. It is so much greater compared to that of like the liver or the pancreas. Now we can look at the structure of tubulin. We see the polypeptide backbone here, and it's this purple structure that is a wire-like structure just winding around through this whole thing. And then we move on to secondary structures, which will consist of the red and the blue features. The helices are the red, and then we have one individual blue piece will be a beta strand, but all together we see it forms this beta sheet. And so the tertiary structures are going to be the beta sheet surrounded by these alpha helices. And so in this image happens to be the ATP form of tubulin, and so there are 3 tertiary structures together which create the quaternary structure. And now we need to be careful not to get that confused, because alpha and beta tubulin are generally regarded to be the tertiary structures for microtubules, because the microtubules are those quaternary structures. And the alpha and beta tubulin are going to bind together in an alternating fashion to create this polar-like structure to form the microtubules for structural support in cells. Here we're looking at the genome of tubulin. This is a very long, complex genome, and in this image it is compared to a pseudogene, and we can see that there are some major differences throughout the whole thing. All the asterisks are going to indicate deletions. On the topic of deletions, we move to mutation. Diseases related to tubulin mutation generally are going to be neurological, and that is because tubulin, beta tubulin in particular, makes up such a large portion of the neurons in our brains. However, that neurological disease is not the only thing that will appear. You'll also have some physical disease, because tubulin is in and of itself a structural protein. So those physical diseases can include facial weakness, scoliosis, pregnancy termination, generally within 3 to 5 weeks, etc. Neurological disease will include Asperger's, ADHD, seizures, temper tantrums, intellectual disability, psychomotor delay, optic atrophy, hypotonia, cognitive impairment, strabismus, epilepsy, and a lot of developmental delays, and there's so many more. In conclusion, proteins are essential. Their shape determines their function, and there are so many different types of shapes that they can take because there are 20 different amino acids. Tubulin is structural, it is not enzymatic. However, enzymes can affect the shape tubulin takes. Alpha and beta tubulin form the microtubules for structural support, and tubulin mutations can cause an array of neurological abnormalities. To close us out, there is some future research potential here. Some future research could distinguish between the intellectual disability and the average intellectual ability by attempting to understand the capacities of this neural network formed by the beta tubulin in the brain. Thank you.
Now, you actually record that this time.
Wait, did you record it or
I
did you
mean,
not?
how do you stop recording? Oh, here.